Okay. First question number 1A, part 1. What is sustainable food production? This question, I made this question from your lesson 1. These points should be included. First one is the production of food. Okay, the production of sufficient amount of food. Okay, the production of sufficient amount of food for the human population. For the human population. For the human population, using environmentally safe methods. Using environmentally safe methods. Okay, it is called as sustainable food production. So these points should be included. The production of sufficient amount of food for the human population using environmentally safe methods. Okay, environmentally safe methods, very important point. Environmentally safe methods. Okay, production of sufficient amount of food for the human population using environmentally safe methods. It is the sustainable food production. Mention 3 steps based on biological knowledge that can be used for sustainable food production. First point, production of high yielding varieties of plants and animals. Okay, first one is production of high yielding varieties of plants and animals. I think you have already written this the answer. If no, please search your resource book. These answers are available in your resource book under the lesson 1. Okay, first one, production of high yielding varieties of plants and animals. The second point, production of disease resistant plants and animal varieties. Production of disease resistant plants and animal varieties. Third one, improve the post-harvest technology methods. Improve the post-harvest technology methods. What do you mean by the post-harvest technology? After harvesting. Okay. After harvesting, what about the transport? What about the how they are arranged in racks in their containers? Okay, how it is done? It is the post harvesting technology. ඒ කියන්නේ crop එක harvest කරාට පස්සේ දැන් පැඩි නම් මොකද්ද ඒක crop කරාට පස්සේ ඒ ටික කොහොමද transport කරන්න අදාළ තැනට නේද ඕනම එකක් අපි හිතමුකෝ potato කියලා potato harvest එක ඒ කියන්නේ potato එකේ potato ටික crop කරලා ගත්තට පස්සේ අදාළ plant එකකින් කොහොමද potatoes ටික shop එකට යනකම් අරගෙන යන්නේ නේද එන්නේ ඒ ටික තමයි post harvest කියලා කියන්නේ post harvest period එක so post harvest technology improve the post harvest technology methods there are 3 steps. Name the type of the tetrose known you. It is the erythrose. Erythrose. That means C4. C4. That means tetrose. Okay. Erythrose are rare in nature. Erythrose sugar is rare in nature. Mention 3 works of cilia and flagella. First one, act as locomotor appendages. Act as locomotor appendages. Second one, can move fluid over the surface of the tissue. Can move fluid over the surface of the tissue. Third one, cilia lining in oviducts. Cilia lining in oviduct. Okay. Can you imagine if this is the ureter? Here the oviduct. Okay. So cilia lining in the oviducts. Cilia lining in the oviducts help move an egg towards the ureter. This is the ovary. Ovary produce the egg. Okay. This is the oviduct. This is the uterus. So these cilia helps to move these eggs towards the uterus. Cilia lining in oviducts help move an eggs towards the uterus. What are the components of the plasma membrane? It should mainly lipids and proteins. That's enough. Carbohydrates are also involved. So what are the components next? So you can write lipids, proteins, and carbohydrates. The main lipid type is phospholipids. Other than that, cholesterols are also involved. Okay, proteins and carbohydrates. What are the peripheral proteins of the plasma membrane? So how these three organic components are contributing the making of plasma membrane? Those are phospholipids. Okay, so lipids are involved. This is the phospholipid bilayer. Okay, protein molecules are embedded through this phospholipid bilayer, and again we can see protein molecules and carbohydrate chains. Okay, these structures called as glycolipids because this carbohydrate chain is attached to the lipid molecule, phospholipid molecule, and here the carbohydrate molecule is attached to the protein, therefore glycoprotein. Okay, and between these two lipid layers, cholesterol molecules also can be seen. So lipids, both are lipids, cholesterol, lipid and phospholipids. Okay, lipid, proteins, mainly these transmembrane proteins, and other than that, if this is the inside, okay, inner environment of the cell, okay, if this is the inner environment, nucleus and everything is there, so we can see peripheral proteins like this. Okay, peripheral proteins. Those are the peripheral proteins. You have to define what are the peripheral proteins. Proteins which are not embedded in the lipid bilayer. You can see these proteins are not embedded within the phospholipid bilayer. Proteins which are not embedded in the lipid bilayer and loosely bound to the inner surface of the membrane. Loosely bound to the inner surface. Okay? This is the outer surface. Okay, this is the outer surface and this is the inner surface. These peripheral proteins are loosely bound to the inner surface of the membrane called peripheral proteins. How we can define proteins which are not embedded in the lipid bilayer and are loosely bound to the inner surface of the membrane called peripheral proteins. Okay, then what are the three types of junctions found in animal cells? First one, tight junctions. Tight junctions. Tight junctions. Second one, desmosomes or anchor. Then desmosomes or anchor junctions and gap junctions or communicating junctions. Okay, first one, tight junctions, desmosomes or anchor junctions, gap junctions or communicating junctions. What is the active site of an enzyme? Okay, if this is the enzyme, always enzyme is larger than the substrate molecule. If this is the substrate molecule, the specific site, this site within the enzyme we call it as the active site. This is the active site. Okay. The substrate binds to the specific region of the enzyme. The substrate binds to a specific region of the enzyme. This region is called as the active site. So that's enough. What is the cooperation? Cooperation under the enzyme part, what is the cooperation? Okay, what is the cooperation? Described in the allosteric regulation of enzymes. How we can define it? So you have to write this manner. Binding of one substrate molecule can stimulate binding or activity at other active site, thereby increase the catalytic activity. It is the cooperation. What is the cooperation? Described under the allosteric regulation of enzymes. Binding of one substrate molecule can stimulate binding or other active site, thereby increase the catalytic activity. It is the cooperation. What is the respiratory quotient? Respiratory quotient means, you know, RQ. RQ is equal to VCO2 over VO2 now. So how we can define it? The ratio here, the ratio of carbon dioxide. Ratio of carbon dioxide evolved carbon dioxide and absorbed oxygen volume. No? The ratio of carbon dioxide evolved and the volume of oxygen consumed or absorbed. Okay, volume of oxygen consumed in a given time for the respiratory substrate. It is the respiratory quotient. The ratio of carbon dioxide evolved and the volume of oxygen consumed in a given time for the respiratory substrate. It is the respiratory quotient. What is the global importance of photosynthesis? Answer is available in your resource book. First one, all life on earth. All are depends on the sun. No? Sun, that means the photosynthetic organisms, that means photosynthesis directly or indirectly. So all life on earth depends on photosynthesis. Directly or indirectly. Second one, fulfill both carbon and energy requirements. For organisms. Third one, provide oxygen for respiration. Of aerobic organisms. Fourth one, maintain oxygen and carbon dioxide balance in the atmosphere. Fifth one, production of fossil fuel. Sixth, maintain the global temperature, because photosynthetic organisms absorb carbon dioxide. Carbon dioxide is the main reason for the atmospheric increasing of the global temperature. So photosynthesis maintains the global temperature by absorbing carbon dioxide. Okay, all the life on earth depends on the photosynthesis, directly or indirectly. Fulfill both carbon and energy requirements for organisms. Provide oxygen for the respiration of aerobic organisms. Maintain oxygen and carbon dioxide balance in the atmosphere. Production of fossil fuel. Maintain of global temperature. Which animal kingdoms have an organized nucleus? I ask about eukaryotic kingdoms. Okay, eukaryotic kingdoms. You know, there are three domains. Domain, archaea, domain, bacteria. These two are prokaryotic organisms. That means their DNA is naked, okay, exposed to the external environment. That means their genetic materials, DNA or RNA, is not enclosed by a membrane. Okay, they are naked. They are open DNA. But in domain eukarya, they have an organized nucleus. Nuclear materials are enclosed by a membrane. Okay, nuclear materials are enclosed by a membrane, so they are eukaryotes. What are the four kingdoms? Kingdom Protista, kingdom fungi, kingdom Plantae, kingdom Animalia, kingdom Animalia. Okay. Name 3 protists which have heterotrophic nutrition. It should 2. According to your syllabus, 2 examples are enough. So, what are the 2 examples? Paramecium and amoeba. Okay. Heterotrophic means they can't produce their own food. They have to depend on other organisms' bodies or other organisms' productions. So they are heterotrophs. What are the 2 heterotrophic protists? Paramecium and amoeba. Under the protista kingdom, we are talking about algae. All algae are photosynthetic. That means all algae organisms are autotrophs. Okay, autotrophs. What are the similar characters of Neem to the angiosperms? You know about the plant classification. Up to now, we so many times we have discussed about this plant classification. How plants are developed. Okay, how they become advanced. So under the presence or absence of the vascular system, mainly plants are divided into 2: non-vascular plants and vascular plants. Non-vascular plants and vascular plants. Non-vascular plants commonly known as bryophytes. Bryophytes, and under the bryophytes we are talking about 3 phylums. Phylum Bryophyta, example Mosses and Pogonatum, and phylum Anthocerophyta. Example is Anthocerus. Third one is phylum Marcanthophyta. Okay, example is Marcanthia. Okay, these 3 phylums haven't a vascular system like xylem and phloem. No vascular tissues. That means they are non-vascular plants. And also in their life cycle, gametophyte is the dominant one. Okay, it is very primitive character. Gametophytic generation, dominant means very primitive character. In all 3 phylums, the dominant plant in their life cycle is the gametophyte. But they become vascular plants. Always its life cycle, the dominant plant is the sporophyte. Okay, sporophytic generation is the dominant generation. So it is an advanced feature. In vascular plants, we divide these vascular plants into 2 categories. First one is what? Seedless plants and seed plants. Okay, seed plants. Seedless plants, we are talking about 2 phylums. Phylum Pterophyta, example is Nephrolepis. And the second one is phylum Lycophyta. There are 2 examples you have to study: Lycopodium and Selaginella. Okay, what about the spores? In here, all bryophytes, homosphoric spores are presented. Homosphoric means their external feature is same to each other, but they are differ in the genetic composition. So homosporous, all are homosporous, all pterophytes are homosporous. Under the phylum Lycophyta, Lycopodium is homosporous, but Selaginella heteros. Heterosporous, that means megaspore and microspores. There are two different spore types, megaspore and microspore. In seed plants, okay, in seed plants, all are heterospores. And seed plants can be categorized into two. First one is gymnosperms. Second one is angiosperms. Okay, angiosperms. Gymnosperms are three phylums. Phylum Cycadophyta. Cycus is the example. Phylum Coniferophyta. Pinus is the example. Phylum Neotophyta. Neatum. Here the neatum. Neatum is there. Okay. So they are evolutionary. There is a close evolutionary relationship between the phylum Neotophyta and phylum Anthophyta. Okay. Here Anthophyta. Angiosperms means phylum Anthophyta. That means flowering plants. Okay, there is a close relationship between phylum Neotophyta and phylum Anthophyta. So there are some features, okay, which can be seen in neophytes related to the anthophytes. Okay, so what are the two similarities? What about their leaves? In neotum, its leaves look like this. Okay, that means their leaves looks like a leaf of dicot. Dicot means phylum Anthophyta, class Monocotyledone and Dicotyledone. So monocot and dicots. Here the neophyte leaf is external feature looks like a dicot leaf. It is the first one. External appearance of leaves are similar to dicot leaves. Okay, external appearance of leaves are similar to dicot leaves. Second one, their seeds, they have no fruits. No, gymnosperms have no fruits, but angiosperms have a fruit. Their seed is enclosed by a fruit neck, but even they haven't a fruit. Their seeds look like a fruit. Okay, so the second feature is their seeds are look like as fruits of angiosperms. Their seeds looks like a fruit of angiosperms. Tabulate the differences between two classes of angiosperms. Here, phylum Anthophyta can be divided into two classes, class Monocotyledone and Dicotyledone. So what are the five, I think, 7 differences, okay? 7 differences. Answers are already, already available in your resource book. So anyway, we have to discuss rapidly. Here, monocots and dicot. Okay, what is the first difference? First one is the embryos have only one cotyledon. One cotyledon, here 2 cotyledons. Okay, the embryos have only one cotyledon. Embryos have 2 cotyledons. Embryos have only one cotyledon. Embryo have 2 cotyledons. Second one is, that is why monocot. Okay, cotyledon, like cotyledon. Therefore, they called as cot. Mono means one. Here di means two. One cotyledon means monocot. 2 cotyledon means dicot. Okay. So the second one is what about their root system? Here fibrous root system. Okay, fibrous root system. Here taproot system. This is the taproot. This is the ground level. This is the taproot, and it is branched into 2 and again branched. Okay, so many times branched. This is the taproot system. So what about their veins? Veins in leaves here in monocots. Veins are parallel to each other. Okay, parallel veins. Parallel veins, and here in leaves, the veins are reticulate. Reticulates like taproot system, like that. Their veins are branched and again branched into two, again branched into two. This one also branched into two, like that. Okay. So reticulate vein. Reticulate veins. Okay. First difference is the embryos have only one cotyledon. Here embryos have two cotyledons. Fibrous root system, taproot system. Parallel veins in leaves, reticulate veins in leaves. Here, flower parts are trimerous. Trimerous means especially petals. Okay, trimerous means three petals. Okay, if you can observe coconut flower, very small, but we can observe it. Only three petals. Okay, so trimerous. Here, pentamerous. Pentamerous, pentamerous or tetramerous. Penta means 5 petals, tetra means 4 petals. Pentamerous or tetramerous. Okay, flower parts are trimerous. Here, flowers are tetramerous or pentamerous. Here, perianth. Perianth present in flowers, that means no distinct calyx and corolla. Here, distinct calyx and corolla in flowers. That means actually this is the calyx. And what about the petals? In shoe flower, we can differentiate the corolla and calyx. But in coconut flower, we cannot separate these things. No, they are attached, like attached. So how we can say it? No distinct calyx and corolla. Here, no distinct calyx and corolla. That means perianth present in flowers. Here, distinct calyx and corolla in flowers. Distinct calyx and corolla in flowers. Here, pollen grains are with one opening or aperture. One opening. Pollen grains are with one openings or aperture. Here, pollen grains are with three openings or three apertures. Okay. 7. Vascular bundles in the stem do not have cambia and scattered. It's look like this. What about the cross section of monocot stem? This is the epidermis, and vascular bundles are scattered. In peripheral region, they are very small and high in numbers. Okay, very small vascular bundles. If we enlarge one vascular bundle, it's look like this. It's look like this. No, if this is the xylem, if this is the phloem, no cambium. Okay, no cambium between xylem and phloem. Sclerenchyma sheath can be seen around the vascular bundle in monocots. So peripheral vascular bundles are very small and high in numbers. When they come into the center region, their size is somewhat large and less in numbers. Okay, anyway, this structure we called as scattered. Okay, vascular bundles are scattered. But in here, vascular bundles in the stem have cambia and arranged as a ring. Okay, it's look like this. If this is the epidermis, vascular bundles are arranged in a ring like this. When we enlarge one vascular bundle, it's look like this. There is the cambium. If this is the phloem, if this is the xylem, this is the cambium. This is the cambium. This is the sclerenchyma cap. This is the sclerenchyma cap. Here, sclerenchyma sheath. Okay, here we cannot see a cambium between xylem and phloem. That is why we cannot see the secondary growth in monocot roots as well as monocot stems. Anyway, xylem and phloem, between xylem and phloem, no cambium, but in here, in dicot stems as well as the roots, between the phloem and xylem, we can see a cambium. Okay, here, in both cases, sclerenchyma tissues are involved to uh, make the vascular bundle. Here, as this is the sclerenchyma tissue. Here, the sclerenchyma cap. Okay. So those are the 7 differences. First one here. Here the embryos have only one cotyledon, fibrous root system, parallel veins in leaves. Flower parts are trimerous. Perianth present in flowers, that means no distinct calyx and corolla. 6. Pollen grains are with one opening, no aperture. Vascular bundles in the stem do not have cambia, are scattered. That means vascular bundles are scattered. This side, embryos have 2 cotyledons, taproot system, reticulate veins in leaves. Flowers are pentamerous or tetramerous, distinct calyx and corolla in flowers. Pollen grains are with 3 openings or apertures. Vascular bundles in the stem have cambia and arranged in a ring. Okay. So, state the characteristic features of flowering plants. What are the characteristic features? First one, stamen produce microspores and these microspores developed into pollen grains. Pollen grains contain male gametophytes or gametes. It is the first point. Stamens produce microspores and these microspores developed into pollen grains. Pollen grains contain male gametophytes or gamete. Second one is carpel, the female part. Okay, carpel produce megaspores. And these megaspores produce female gametophytes or embryonic sacs. Third one, seeds are enclosed within carpels. Fourth one, production of fruits. What is the monomer of common stored food in fungi and animals? The common stored food is glycogen. The monomer of glycogen is glucose. Mention 2 main principles of Lamarck theory. First one, use and disuse. Second one, inheritance of acquired characteristics. First one, use and disuse. Second one, inheritance of acquired characteristics. Second question A. Okay, second question A. What is cardiac cycle? How much time will it take? Cardiac cycle means, you know, in the cardiac cycle, contractions and relaxation of ventricles and arteries are involved in the cardiac cycle. It's look like this. The full time is 0.8 seconds. Here 0.4 seconds and 0.3 seconds, 0.1 seconds. No? So what is the cardiac cycle? The sequence of events that takes place in a complete heartbeat. Mention the phases of cardiac cycle with the corresponding time periods. There are 2 systole. Systole means contractions. Systole means contractions. Diastole means the relaxation. No. So first one is this represent atrial systole. That means atrial contraction. Okay, you had to write atrial systole. The relevant time is 0.1 second. The next one is ventricular systole. This is the first one. This is the second one. Initially atria, 2 atria should be contracted. Then 2 ventricles should be contracted. As the third step, both should be relaxed, no? So second one is ventricular systole. It is 0.3 seconds. That means ventricular contraction. Ventricular contraction. Here, complete cardiac diastole. Complete cardiac diastole. It is both atria and ventricles relaxation. Complete atria and ventricle relaxation. Complete cardiac diastole. So your answer. First one is atrial systole. Initially, the heart two atria should be contracted. The time is 0.1 seconds. Mulima atria contract vela ventricles වලට blood supply කරන්න ඕනි. ඊට පස්සේ ventricles වල තියෙන blood two arteries here, pulmonary artery and aorta. නේ? එතකොට pulmonary artery එකටයි aorta එකටයි pump කරන්න ඕනි. එතකොට ventricles contract වෙන්න ඕනේ. ඊට පස්සේ මොකද වෙන්නේ? atria වගේම ventricles relax වෙන්න ඕනේ. එතකොට තමයි වෙනා කාවා වලින් නේද? both වෙනා කාවා වලින් මේ right atria එකට blood ටික එන්නේ deoxygenated blood. එතකොට pulmonary veins වලින් left atria වලට oxygenated blood එන්නේ. මේක තමයි relaxation period එක. Okay, atrial systole, that means 0.1 seconds, ventricular systole 0.3 seconds, complete cardiac diastole 0.4 seconds. Name the instrument for measuring atrial blood pressure. Sphygmomanometer. Sphygmomanometer. 9 factors affecting hypertension. Hypertension means high blood pressure, like that. Okay. ඒක ලොකු time duration කියන්නේ ලොකු කාලයක් තිස්සේ එන high blood pressure තමයි hypertension කියන්නේ. Tension එක කියන්නේ pressure එක. Blood pressure means the pressure created within the blood vessel. නේද? blood vessel එකක blood යනකොට ඒ blood වලින් මේ අදාළ vessel එකේ walls වලට දෙන force එක. force area එකකින් divide කරාමනේ pressure එක එන්නේ. එතකොට මේ blood වලින් මේ wall එකට දෙන force එක තමයි එක 1 square meter වගේ area එකකට දෙන force එක කියලා හිතන්නකෝ. ඒක තමයි pressure එක. එතකොට pressure එක වැඩි වුණාම මොකද්ද? high blood pressure වෙනවා. ඒ high blood pressure ගොඩක් කාල තියෙනකොට hypertension වෙනවා. So mention 9 factors affecting hypertension. First one, obesity, diabetics, family history, smoking, a sedentary lifestyle, කම්මැලි ජීවිතයක්, high intake of salts, high intake of alcohols, stress, deposition of low density lipoprotein on artery walls. That means LDL, bad cholesterol. Okay. Obesity, yani මහත් ස්ථූලතාවය කියන එක. Obesity, diabetics, දියවැඩියාව, family history, smoking, a sedentary lifestyle, high intake of salts, high intake of alcohols, stress, deposition of low density lipoprotein on artery walls. Define single circulation and double circulation. කොහොමද single circulation එකේදී වෙන්නේ double circulation එකේදී වෙන්නේ? Circulation or circulatory systems can be divided into two. Circulatory system. ඔයාලා මොනවා හරි දෙයක අමාරු පාර්ට් එකක් තියෙනවා නම් ඒකේ අනිවාර්යයෙන්ම කැටගරයිසේෂන් එකක් ඒකේ හයිඩ් වෙලා තියෙනවා ඒක ඇතුලේ. ඒ හයිඩ් වෙලා තියෙන කැටගරයිසේෂන් එක හරියට අල්ලගෙන එතනින් හිතන්න පටන් ගන්නවා. හරි circulatory systems can be divided into two. Open circulatory system and closed circulatory system. No? Open circulatory system means blood is not always restricted to a vessel. Here, blood is always restricted to a vessel. Then, heart එකේ එළියට ගියපු blood ටික ආපහු heart එකට එන කම්ම යන්නේ vessels through. ඒකට ඒක තමයි closed circulatory system කියලා කියන්නේ. Closed circulatory system can be divided into two: single circulation and double circulation. No? Single circulation and double circulation. In single circulation, the chambers of the heart is always two chambered heart. Here, three chambered or four chambered heart can be seen. Okay, two chambered heart means one artery, and one ventricle. We can see such a circulation in fish, both cartilage and ostic fish. Okay. In double circulation, a heart may be three chambered. That means incomplete wall of between two ventricles. Especially in amphibians and reptiles. But not all reptiles, like alligators and crocodiles, we can see four-chambered heart. In four-chambered heart, ventricles and arteries are fully separated, and two ventricles and two arteries are fully separated. So in single circulation එකේදී වෙන්නේ body එක through එකපාරක් යනකොට එකපාරක් blood යනකොට මේ body එක through එකපාරක් blood යනකොට මේ පැත්තට body එක through එකපාරක් blood යනකොට heart එක through එකපාරයි. It is the single circulation. හැබැයි මෙතනින් වෙන ආකාර වලින් blood එනවා deoxygenated blood. එතකොට එන්නේ අයෝටා එකෙන් රිලීස් කරපු oxygenated blood තමයි tissues වලට ගිහිල්ලා නේද deoxygenated වෙලා එන්නේ. එතකොට body එක through එකපාරයි දැන් ඔන්න blood මෙතන flow වෙන්නේ. මේ body එක through එකපාරයි blood flow වෙන්නේ. එතකොට මෙතනින් right atria එකෙන් right ventricle එකට එකපාරක් යනවා. ඔන්න one time. And මෙතනදී pulmonary arteries create වෙලා නේද? Pulmonary arteries create වෙලා pulmonary arteries වලින් lungs වලට ගිහිල්ලා deoxygenated blood කරලා ආපහු pulmonary veins වලින් heart එකට අරන් එනවා right ventricle right atria එකට. එතකොට එතනින් ආපහු left ventricle එකට heart blood pump කරනවා. එතකොට two times යනවා heart එක through නේද? body එක through one time නම් heart එක through two times. එතකොට එතන double circulation. So how we can explain? In single circulation, in a complete circulation through the entire body, blood passes through the heart only once. But But in the double circulation, in a complete circulation through the entire body, blood passes through the heart twice. Explain the physical and chemical digestion of food in the oral cavity. What about the food digestion of oral cavity? Which type of foods are chemically digested within the oral cavity? So if this is the oral cavity, if this is the nose and this is the opening, this is the oral cavity, and this is the pharynx, and this is the esophagus. No? Pharynx is a common chamber for both respiratory tract and digestive tract. Here also due to respiratory tract. So this is the pharynx, and this is the esophagus, a tubular structure, and this is the stomach, and this is the duodenum, very first part of the small intestine. This is the small intestine, and this is the large intestine. Your digestive system looks like this, no? Rectum and anus, no? In the oral cavity, this is the oral cavity. Both mechanical digestion, chemical digestion, enzyme is presented in saliva, that means in the oral cavity. In the oral cavity, by using the amylase enzyme, starch is digested into maltose. So, chemical digestion also happening within the oral cavity. Carbohydrate digestion, that means polysaccharide digestion. You know, starch is a polysaccharide. Maltose is a disaccharide. Okay, polysaccharide and disaccharide. So you have to explain physical or mechanical digestion by using teeth. Using teeth, muscle contraction of tongue and palate. Food is broken into small particles. Mechanical digestion එකේදී food එක small particles වලට break වෙනවා. එතකොට surface area එක වැඩි වෙනවා. Chemical digestion එක සිද්ධ වෙලා يعني enzymes වලට මේ හරි involvement surface area එක increase කරන එක තමයි mechanical digestion එකේදී වෙන්නේ. Okay? Using teeth, muscle contraction of tongue and palate, food is broken into small particles. That means increase the surface area for the action of enzymes. Increase the surface area. Chemical digestion by using amylase enzyme, starch, that means a polysaccharide, is digested into a disaccharide. That means starch is digested into maltose. Okay. Gastric juice consists of what? Gastric juice consists of, first one, mucus, pepsinogen, the inactive form of pepsin, pepsinogen and HCl. Mucus, pepsinogen and HCl. Okay, mucus, pepsinogen, HCl. Mention three main layers of the heart wall. Pericardium, the outermost layer is the pericardium. Second one is the myocardium. Muscles actually. Cardiac muscles are presented in the myocardium. Conducting system of the heart also presented in the myocardium. Therefore, myocardium is very important. The outermost layer is pericardium, the middle layer is myocardium, the innermost layer we call as endocardium or epicardium. So you have to write endocardium. Okay, paper epicardium endocardium. Pericardium epicardium paper epicardium you have to understand this is the pericardium. Within the brackets you can write epicardium, pericardium, myocardium and endocardium. What are the parts of conductive system of the heart? First one, SA node, that means sinoatrial node. No, second one is AV node, that means atrioventricular node. Third one, atrioventricular bundle. This is the heart. We can see the AV node, SA node. This is the SA node. It generates impulses in this manner. This is the intraventricular wall. We can see the AV node. Okay, AV node, and this is the what? This is the atrioventricular bundle, that means bundle of His, and it is divided into two bundle branches, and they are divided further. We called as Purkinje fibers. Okay. SA node or sinoatrial node, AV node or atrioventricular node. Third one, atrioventricular bundle, that means bundle of His, bundle branches and Purkinje fibers. Give another name for plant growth regulators. Plant hormones. Other name is plant hormones for plant growth regulators. Mention four other functions of growth regulators that delay leaf senescence in plants. What are the another functions? First one, regulate cell division in shoots and roots. Second one, modify apical dominance and promote lateral bud growth. Third one, promote movement of nutrients into zinc tissues. Fourth one, stimulate seed germination. Fifth one, delay leaf senescence. But no need to write it again. Regulate cell division in shoot and roots. Modify apical dominance and promote lateral bud growth. Promote movement of nutrients into zinc tissues. Stimulate seed germination. Mention 8 trace elements obtained from the soil solution by the root of plants. What are the 8 trace elements? Essential elements are 2 types, macro elements and micro elements or trace elements. I ask about the elements. Okay, chlorine. Chlorine is taken as chloride from soil solution, and iron as Fe2 plus. Okay, then zinc as zinc 2 plus. Okay, all are taken as the from the soil solution. This is the fourth one is boron, H3BO2. Sorry, H2BO3 minus. H2BO3 minus iron. And copper as copper 2 plus. Copper 2 plus. And molybdenum. Copper 2 plus or copper plus. Then molybdenum MOO4 2 minus. Nickel 2 plus. And manganese 2 plus. Okay, those are the 8 trace elements: chlorine, iron, zinc, boron, copper, molybdenum, nickel and manganese. මෙතනට ලියන්න ඕනේ නෑ මේ ටික. නමුත් ඔයාලා ලියා ගන්න soil solution එකකින් මේගොල්ලන් absorb කරන්නේ chlorine නම් chloride ion විදිහට. Here Fe2 plus විදිහට, zinc 2 plus, H2BO3 minus, copper 2 plus or copper plus, MOO4 2 minus, nickel 2 plus and Mn 2 plus. Give 5 differences between hardwood and sapwood. Next question, differences between hardwood and softwood. Okay. hardwood පටල ගන්න එපා. hardwood එකයි sapwood එකයි කියලා කියන්නේ එකම ගහ එකම plant එකේ තමයි මේ දෙක තියෙන්නේ. හරිද? hardwood එකයි sapwood එකයි. ඒකට hard කියලා කියන්නේ මැද්ද. මැද්ද තියෙන වගේ අදහසක් නේ. එතකොට මේ තියෙන්නේ hardwood එක. hardwood එක මේ තියෙන්නේ sapwood එක. So what are the differences? First difference is here non-living. Hardwood is non-living. Here living part. Okay, non-living and living. Here the second is situated at the center of the wood. Situated at the center of the wood. Okay. Here situated at the peripheral region. Situated at the peripheral region of the wood. Third one is dark in color. Dark in color. Dark in color. Here light in color. Here thickness is comparatively high. Thickness is comparatively high. Here it is comparatively soft structure. And tyloses or teloses. Tyloses present, tyloses absent. Those are the 5 differences. What about the hardwood and softwood? Hard. Here hard, then hardwood and softwood. Okay. Hardwood means the wood of angiosperms. Softwood is the wood of gymnosperms. Wood of gymnosperms. Wood means secondary xylem. No? Here the secondary xylem of angiosperms. Here also comparatively dark in color. Okay, comparatively dark in color. ඔයගොල්ලන් සාමාන්‍යයෙන් ගෙවල් වල ලී බඩු හදන්නේ මේ hardwood එකකින්. හැබැයි සමහර වෙලාවට අපිට showrooms එකේම අර බර අඩු මේ එච්චරම ලොකු වටිනාකමක් නැති නිකන් කාඩ්බෝඩ් වගේ ලී තියෙන්නේ වුඩ් එක තියෙන්නේ ඒක තමයි සොෆ්ට් වුඩ් කියලා කියන්නේ. ගොඩක් වෙලාවට ඔය කම්පියුටර් ටේබල්ස් වල හිම හදලා තියෙන සමහර නිකන් අර ලයිට් කලර් එකට ඒකේ ඔරිජිනල් කලර් එකත් ලයිට්. ඒ කියන්නේ මේ ලොකු ඩාර්ක් කලර් එකක් නෑ. අපි සාමාන්‍ය ගෙවල් වල සෝෆා එම ඒ මොනවාද මේ බෙඩ් එක ඒක ඔක්කොම හදන්නේ හાર્ඩ් වුඩ් එකෙන් සොෆ්ට් වුඩ් යූස් කරලා තමයි අර වගේ දේවල් හදන්නේ. එතකොට මෙතනදී comparatively dark in color, comparatively dark in color, comparatively light in color. Second one is weight. Weight is comparatively high. උස්සන්න අමාරුයි හොඳ ලී වලින් හදලා තිබ්බොත් hard wood එකෙන් හදලා තිබ්බොත්. හරිම මේ ඒ කියන්නේ බර අඩුයි. මෙතනදී weight එක අඩුයි. light weight එකක් තියෙනවා. weight එක අඩුයි. And high expensive. High expensive, low expensive. Low expensive. Fourth one is xylem vessels are present. Xylem vessels are present. Xylem vessels are absent. Fifth one is this is the secondary xylem of angiosperms. Here the secondary xylem of here the wood of gymnosperms. No? Wood of gymnosperms. Secondary xylem of angiosperms. Okay, I think you have completed. Here comparatively dark in color. Weight is comparatively high. High expensive. Xylem vessels are present. This is the secondary xylem of angiosperms. Here in softwood, comparatively light in color, comparatively low in the weight, low expensive. Xylem vessels are absent. This is the wood of gymnosperms. Draw two cross-sectional diagrams to show the appearance of dicotyledons before and after secondary growth. We use a dicot stem. What about the primary structure? That means before the secondary growth. What about the primary structure? Primary structure looks like this. This is the epidermis. This is the epidermis. And those are the vascular bundles. Vascular bundles, and this is the vascular cambium. Vascular cambium, and this is the epidermis, this is the cortex, and this is the teeth. This is the vascular region. No? Vascular region. As the inner part, we can see the primary xylem. This is the primary phloem. This is the primary phloem. This is the before secondary growth, you had to draw this structure, and after the secondary growth, you have to mention about two cambiums. Okay, secondary growth means increase the diameter and kanda mahatin වැඩි වෙන එක. කඳ හරි මුල හරි root එක හරි root එක හරි මහතින් වැඩි වෙන එක තමයි secondary growth එක කියන්නේ. එතකොට if this is the cork cambium, what would be the peripheral tissue layer? This is the cork. Right? If this is the cork cambium, this is the cork. Innermost layer is secondary cortex, but you no need to label it because your resource book hasn't labeled it. So this is the cork cambium. This is the cork, and this is the vascular cambium. This is the vascular cambium. This is the vascular cambium, and this is the vascular cambium. This is the secondary xylem. secondary xylem. Okay, this is the secondary xylem, and you can see primary xylem in this manner. This is the primary xylem. Okay, this is the vascular cambium. This is the secondary phloem. Secondary phloem, and those are the primary phloem. This is the primary phloem. Okay, race looks like this. Vascular race looks like this. Vascular race looks like this. Mention two main types of sclerenchyma cells: sclerides and sclerenchyma fibers. Sclerides, sclerenchyma fibers. Which tissue in plants contains cells that cannot divide further? Parenchyma, sclerenchyma, collenchyma, xylem, phloem, epidermis. Okay, all 6 tissues should be included. Can you remember the plant tissue classification? I think you can remember because so many times we have discussed in previous papers. So anyway, I will mention plant tissues. Plant tissues can be divided into 2. Okay, first one is dividing cells. That means meristems. Okay, the second one is non-dividing. That means they are differentiated to a particular function. Meristems are 3 types. That means dividing tissues or meristems: apical meristem, lateral meristem, and intercalary meristem. Now, differentiated or non-dividing tissues can be divided into 2: simple non-dividing means permanent. We can see also permanent. Simple permanent or simple differentiated tissues, and complex differentiated tissues. Okay, simple means the tissue is composed of single types of cells. Okay, more than one type of cells. Okay, what are the 3 simple permanent tissues? Parenchyma, sclerenchyma, and collenchyma. Okay, complex permanent tissues. First one is xylem tissue, phloem tissue, and the okay. Which tissues in plants contain cells that cannot be divided further? That means non-dividing or permanent tissues or differentiated tissues. So parenchyma, sclerenchyma, parenchyma tissue, sclerenchyma tissue, and collenchyma tissue. Xylem tissue, phloem tissue, and the epidermis. Mention 3 types of tissue containing systems you mentioned above. That means the tissue systems, 3 tissue systems can be applied into the differentiated tissues, because this tissue system is composed of differentiated for a particular function. Therefore, we have to apply these 3 the tissue systems into this region. These 3 represent the ground tissue system. It is the first tissue system, ground tissue system, and the second one is vascular tissue system, and the third one is dermal tissue system. Dermal tissue system. Okay. Ground tissue system, vascular tissue system, and dermal tissue system. Okay. Which reagent is used to identify starch in science lab? Iodine solution. Iodine solution. What would be the observation of above experiment? Dark blue coloration. Starch. So the starch, identification of starch, we use iodine solution. Iodine is yellowish brown in color. After adding I2 into starch, the positive observation is dark blue coloration. Which two solutions should be used in addition in the laboratory experiment in which the activity of enzymes is studied using the test in one above. This is the action of amylase enzyme, no? Identification of the action of amylase enzyme. Initially, we have to prepare two solutions. First one is the starch solution. Second one is amylase solution. Now we have to mix with each other. That means starch plus amylase. මෙතනදී mix කරපු ගමන්ම අපි stop watch එක start කරනවා. ඒ වෙලාවේ stop watch එක start කරපු වෙලාවෙම මෙතන reaction එකක් මුකුත් වෙලා නැහැ. That means starch amylase වලින් maltose වෙනවනේ. මෙතනදී මේ mixtures එක මේ solutions එක mix කරපු ගමන්ම immediately මේ reaction එක වෙන්නේ නැති හින්දා මේකෙන් drop එකක් අරගෙන tile එකකට white tile එකකට දාලා ඒකට iodine දාලා බැලුවා අපිට බලා ගන්න පුළුවන් dark blue coloration. හරිද? 2 minutes වලින් පස්සේ එහෙම මේකෙන් ගන්නවා I2 දානවා බලන්න. එහෙම බැලුවොත් මොකද? Intensity, color intensity is decreased because the action of the maltose after 20 minutes මෙතනදී කිසිම dark blue coloration එකක් එන්නේ නැහැ. That means the reason is all starch molecules are converted into maltose by amylase enzyme. So after adding I2 we can see only the yellowish brown color, which is the color of the iodine solution. So which two solutions should be used in addition laboratory experiment in which the activity of an enzyme is studies? Again, amylase solution and starch solution. Answer is amylase solution and starch solution. Improve the experiment you have mentioned in 2 and explain what your observation is at temperatures of 5 degrees Celsius, 27 degrees Celsius, 40 degrees Celsius and 60 degrees Celsius in the experiment that studies the activity of an enzyme with the temperature. මෙතනදී අපි කියන්නේ constant temperature එකක් තියාගන්න මේ mix කරන එක අපි water bath එකක් use කරනවා. ඒ water bath එක 5 degrees Celsius කරලා මේ experiment එකම එකපාරක් කරනවා. ඊට පස්සේ මේ water bath එක සාමාන්‍යයෙන් 27 degrees Celsius, that means the room temperature එකට ගෙනහලා මේ දෙක mix කරලා ආයෙ මේක observations බලනවා. ඊට පස්සේ අපි මේක 40 degrees Celsius වලට අරන් බලනවා. Optimal temperature එක වෙන්නේ enzymes වල 40 degrees නේ. එතකොට ඒ 40 degrees වලදී amylase enzyme එක denaturate වෙනවා. එතකොට action එක නැති වෙනවා. එතකොට මේක 60 degrees Celsius වලට යනකොට amylase එකේ action එකක් නැහැ. Positive observation එක තමයි 40 degrees Celsius වලට යනකම් temperature එක වැඩි කරනකොට මේකේ color disappear වෙන එක dark blue coloration එක disappear වෙන එක increase වෙනවා. ඒ rate එක increase වෙනවා. But after passing 40 degrees Celsius the rate becomes decreased. හරි. එතකොට ඒ මොකද amylase enzyme එක denaturate වෙන හින්දා. So you have to write when the temperature is increased, when the temperature is increased from 5 degrees Celsius to 27 degrees Celsius to 40 degrees Celsius, the decreasement rate of the blue color intensity is increased. The decreasement rate of the blue color intensity is increased. After passing 4 degrees Celsius. Can you remember the this graph? If this is the temperature in degrees of Celsius, this is the enzymatic rate. Okay, it is not symmetrical, it is an asymmetrical one. This is the optimal temperature. Optimal temperature. After passing optimal temperature, the rate becomes low. No? That means proteins are denaturated after passing. Here, see 0 degrees Celsius, 20 degrees Celsius, and 40 degrees Celsius. Okay, and after passing 40 degrees Celsius, proteins are denaturated. So, when the temperature is increased from 5 degrees Celsius to 27 degrees Celsius, then 40 degrees Celsius, the decreasement rate of the blue color, intensity is increased. After passing 40 degrees Celsius, when the temperature increases, the blue color intensity is increased. The blue color intensity is increased. Got a reason to explain the enzyme. Amylase is an enzyme. Enzymes are made up of proteins. After passing 40 degrees Celsius, enzymes are denaturated. Then what happened? The color intensity is increased. Mention the function of epidermis in plants. First one, as is already available in your resource book, defense against physical damage and pathogens. Defense against physical damage and pathogens. First one, cuticle helps to prevent water loss. Second one, cuticle helps to prevent water loss. Root hairs involved in absorption of water and mineral ions. Root hairs involved in absorption of water and mineral ions. Guard cells help gaseous exchange. Guard cells help gaseous exchange. Hair-like trichomes reduce water loss. Hair-like trichome reduce water loss. Shiny hairs reflect excess light. Shiny hairs reflect excess light. Some trichomes secrete chemicals. Some trichomes secrete chemicals involved in defense against insects. Defense against insect. Pathogens for herbivores. Okay. Have you written? First one, defense against physical damage and pathogens. First one, defense against physical damage and pathogens. Second one, cuticle helps to prevent water loss. Cuticle helps to prevent water loss. Third one, root hairs involved in absorption of water. Root hairs involved in absorption of water and mineral ions. Guard cells help to gaseous exchange. Guard cells help gaseous exchange. Hair-like trichomes reduce water loss. Hair-like trichomes reduce water loss. Shiny hairs reflect excess light. Shiny hairs reflect excess light. Some trichomes secrete chemicals involved in defense against insects, pathogens, or herbivores. What are the main parts of basic structural unit of the nervous system? It is the neuron cell. Okay, neuron. What are the three compartments? Cell body, motor neuron. If this is the cell body of the motor neuron, they are dendrites. No? Dendrites. This is the nucleus. Those are the dendrites, and this is the axon. Axon are branched into two and produce axon bulbs. So you have to write these three. First one is the cell body, dendrites, and the axon. Okay. cell body, then writes axon. Mention all the function of glial cells or neuroglia cells. Okay, glial cells also known as neuroglia cells. No, mention all functions of glial cells by mentioning another name. First one, nourishment of nerve cells. Nourishment of nerve cells. Second one, insulation of nerve cells. Insulation of nerve cells. Third one, replenishing neurons. Replenishing neurons. Fourth one, sometimes modulate neuron functions. Sometimes modulate neuron functions. Okay. State the differences between simple columnar epithelium and pseudostratified columnar epithelium. Simple columnar epithelium, intestinal lining. Simple columnar epithelium, intestinal lining. Pseudostratified columnar epithelium, nasal passage and trachea. Nasal passage and trachea. Give three common functions of epithelial tissues: protection, secretion, and absorption. Protection, secretion, absorption. Okay. Question number 3A1. Give a classification of the human nervous system as you have learned. According to your resource book, the classification should be like this. Human nervous system can be divided into two. Peripheral nervous system, make up peripheral nervous system and central nervous system. Peripheral nervous system and central nervous system. Central nervous system, brain and spinal cord. Brain and spinal cord. Peripheral nervous system, cranial nerves, ganglia outside central nervous system, and spinal nerves. Cranial nerves, ganglia outside central nervous system, and spinal nerves. Which fluid fills ventricles of the brain? Cerebrospinal fluid. Cerebrospinal fluid. Cerebrospinal fluid. Okay, what are the two main parts of the autonomic nervous system? Sympathetic division and parasympathetic division. Sympathetic and parasympathetic. What are the three components of peripheral nervous system? Cranial nerves, ganglia outside the central nervous system, and spinal nerves. What are the types of taste sensed by the human tongue? Sweet, sour, bitter, salt, umami. Sweet, sour, bitter, salt, umami. Umami means the taste of tomato. What are the two types of heat detectors contained in the skin? Bulb of Krauss, organ of Ruffney. Bulb of Krauss, organ of Ruffney. What are the two main functions of the human ear? Hearing and equilibrium. Hearing and equilibrium. Write the two hormones secreted by the posterior pituitary. Posterior pituitary. Produced by the hypothalamus and released. It should not secreted, it should released. Okay, these three correct. It should not secrete, it should released. Hormone secretion or production. Production and secretion කියන එක නිකන් සමානයි වගේ hormones වලදී. So release වෙන එක තමයි posterior pituitary වෙන්නේ. Secretion එකයි කියන්නේ production එක වෙන්නේ hypothalamus. What are the two hormones? ADH and oxytocin. ADH means antidiuretic hormone. Antidiuretic hormone or ADH. Distal convoluted tubule. Target sites ne distal convoluted tubule of the nephrons. Distal convoluted tubule of the nephrons and collecting ducts in the kidney. Collecting ducts in the kidney. Oxytocin, mammary gland and uterine glands. Mammary glands, uterine glands. Mention separately the secretions of the exocrine part and endocrine part of pancreas. Endocrine part, pancreatic juice. Exocrine part, pancreatic juice. Endocrine part, hormones, insulin and glucagon hormones. What are the factors affecting hypothyroidism? Hypothyroidism. Insufficient secretion of thyroid hormone. Insufficient secretion of thyroid hormones, that means T3 and T4 from the thyroid gland. Insufficient secretion of thyroid hormones, that means the T3 and T4 from the thyroid gland. What is the main characteristic of diabetics? The increase in blood glucose level, above the normal limits. What are kidney stones? Kidney stones කියන්නේ precipitation of urinary constituents, usually oxalates and phosphatase in urine. Precipitation of urinary constituents, usually oxalates and phosphates in urine. What are the main steps in urine formation cycle? Ultrafiltration. First one is ultrafiltration. Second one, selective reabsorption. Secretion. Ultrafiltration, selective reabsorption, secretion. Which two types of capillaries from the efferent aorta leaving the glomerulus? Peritubular capillary network. First one is peritubular capillary network. Second one is vasa recta. What are the immunodeficiency diseases? Immunodeficiency diseases means a disorder in which responses of the immune system to antigens are defective or absent. A disorder in which responses of the immune system to antigens are defective or absent. Name two animals which contractile vacuoles are used for the osmoregulation. Amoeba and paramecium. Define antibodies. Proteins secreted by plasma cells. Within the brackets, you have to write differentiated beta lymphocytes. Lymphocytes means white blood cells. Differentiated beta lymphocytes in response to specific antigens. Proteins secreted by plasma cells, that means differentiated beta lymphocytes in response to specific antigens. What are the internal mechanisms of innate immunity? Phagocytic cells. Phagocytic cells, natural killer cells, antimicrobial proteins, inflammatory responses. Phagocytic cells, natural killer cells, antimicrobial proteins, inflammatory responses. Which microorganisms contributes at Mycobacterium tuberculosis. Mycobacterium tuberculosis. Mycobacterium tuberculosis, scientific name. Make sure to write the very first letter in capital and other words are simple. There is a gap between the genetic name and species name. Okay. Mycobacterium tuberculosis. What is the pedigree analysis in the genetics? Diagrammatic representation of the inheritance of a particular trait. Diagrammatic representation of the inheritance of a particular trait within a given family tree. Is called pedigree chart. How to prepare a pedigree analysis? By collecting data for many generations. Within a given family, so that the pattern of inheritance can be understood. Key is essential in reading a pedigree analysis. Show the following terms with symbols in the pedigree analysis. A healthy man, symbolic, a healthy man looks like this. A healthy woman like this. Okay, affected males like this. Affected woman like this. Marriage between two healthy people, marriage looks like this. Okay, imagine they are married and they make two babies, two girls. It represents look like this. Two males, two females. So these two represents the two daughters of this couple. Healthy male and healthy woman. Okay. What are the breeding techniques used in agriculture? First one, artificial selection, inbreeding and outbreeding. Breeding techniques. Breeding techniques used in agriculture. First one, artificial selection. Inbreeding and outbreeding. Third one, hybrid breeding. Intraspecific breeding. And what are the genetic principles? Genetic principles. First one, polyploidy, mutation breeding, third one, genetic modification. Polyploidy, mutation breeding, genetic modification. What is the genetic flow? It should genetic flow. It should not genetic drift. It should genetic flow described in Hardy-Weinberg equilibrium. Okay. Individuals moving in and out of the population. From the population. Individuals moving in and out of the populations may cause appearance of new genes. And disappearance of existing genes. Emigration and immigrations වලදී genes එළියට යනවා, genes ඇතුළට එනවා. ඒක තමයි ඒ කියන්නේ. ඒක තමයි gene flow එක කියලා කියන්නේ. Individuals moving in and out of the populations may cause appearance of new genes and disappearance of existing genes. This is called gene flow. What are sex-linked genes? Sex-linked genes කියලා කියන්නේ certain characters of humans are carried on the certain characters of humans certain characters of humans are carried on the genes, located on the sex chromosomes, Those genes located on the sex chromosomes are called sex-linked chromosomes. Sex-linked chromosomes. What is transformation described in molecular biology? Transformation කියලා කියන්නේ මොකද්ද? Direct uptake of exogenous DNA. Direct uptake of exogenous DNA from the surrounding of a host through its cell membrane and incorporation resulting in a genetic alternation. Direct uptake of exogenous DNA from the surrounding of a host through its cell membrane and incorporation resulting in a genetic alternation. What are the techniques used in recombinant DNA technology? Okay. First one, isolation of DNA from different sources. Okay, isolation of DNA from different sources. Second one, restriction digestion of isolated DNA with restriction enzymes. Separation of DNA fragments by gel electrophoresis. Identification of the correct fragments with desired nucleotide sequences using probes. Joining the DNA fragments from multiple sources using DNA ligase. Joining the DNA fragments from multiple sources using DNA ligase. Okay, shall I repeat again? Isolation of DNA from different sources, restriction digestion of isolated DNA with restriction enzymes, separation of DNA fragments by gel electrophoresis, identification of the correct fragments with desired nucleotide sequences using probes, joining the DNA fragments from multiple sources using DNA ligase. Okay. What is DNA? It should not marking, it should labeling. So please recorrect it. What is DNA labeling? A modification of the DNA strand in such a way that it gives a signal which enables the detection of that DNA strand. A modification of the DNA strand in such a way that it gives a signal which enables the detection of that DNA strand. Complete the following about the tropical lowland rainforest of Sri Lanka. Average annual temperature 28 degrees Celsius. Precipitation 2000 millimeter to 5000 millimeter. What are the three types of ecological pyramids? First one, pyramid of energy. Second one, pyramid of number. Third one, pyramid of biomass. Energy pyramid, number pyramid, biomass pyramid. What are the types of biomes found in the world? What are the biomes? Tropical forest, savanna, desert, chaparral, temperate grassland, temperate broadleaf forest, northern coniferous forest, tundra. 8 biomes we are talking about. tropical forest, savanna, desert, chaparral, temperate grassland, temperate broadleaf forest, northern coniferous forest, tundra. What water conservation adaptations are found in deserts? First one, succulent plant body. Second one, most plants have C4 pathway of photosynthesis. Third one, deep root in shrubs. Fourth one, ability to tolerate heat and desiccation. Fifth one, reduce surface area of leaves. Six, presence of spines or thorns. Seven, presence of toxins in leaves. Okay. What are the trophic levels in an ecosystem? Okay. What are the trophic levels? The producers and consumers. Okay. The producers and consumers in ecosystem can be arranged into several feeding groups. Each known as trophic level or feeding level. The producers and consumers in ecosystem can be arranged into several feeding groups, each group known as trophic level or feeding level. What are the types of trophic levels found in ecosystem? Primary producers, primary consumers, secondary consumers, and tertiary consumers. What are the threats facing biodiversity? First one, habitat loss or fragmentation. Second one, overexploitation. Third one, environmental pollution. Fourth one, introduction of invasive alien species. Fifth one, climate change. What are the keystone species? Keystone species means species that play a very important role in the stability and functioning of a system. Species that play a very important role in the stability and functioning of a system. It is the keystone species. So that's all about the structured essay part. I will send you the essay marking scheme. So I will stop for now. Thank you for joining.